Hey, sorry about the wait. I had to make myself an emergency coffee so I can stay awake. Oh, amazingly, I got through my entire undergraduate degree without drinking coffee. I still don't understand how I did that. Okay, so let's get back to business. Okay, so kind of building on what I put up on the little mini whiteboard. Bloody dog. Let's just bring up the chair. Yeah, I just don't understand how people can live without coffee, to be honest. It's I can't live without it, that's for sure. Um, yeah, as you can see, it depends on what strand we're talking about as to whether or not the strand going left to right is the coding or the template strand, and vice versa on the other way. Right, so DNA is bidirectional. You can have information going in both directions. Pardon me. All you have to remember is that whatever the strand the information is encoded on, it's the other strand which is used as the template. Okay, hopefully that makes sense for everybody. And I'm sorry, Dipper and Galen, for your lack of coffee receptiveness. What about Red Bull? Ah, okay, good point. Dipper, give me a sec. Oh man, you would have thought my typing would be better by now. So it's bidirectional. So each strand. Oh, I'm sorry about that. That's tough. So that's basically what I was saying earlier, Dipper. And so you can actually have, even have genes overlapping, which is kind of rare, but it's not uncommon. Or it's not impossible rather. But you can also have genes which are kind of inverted in a sense, right? So you have gene on one strand, one coding strand going that way, a gene on the other strand going that way, and they actually can share a regulatory region, right? And so a couple of the genes that I've come across in my own research act as pairs. And so that's really interesting because that kind of suggests that there's a common mechanism of turning both genes on or off, right? And so that's super interesting when we think about control of gene expression. Okay, so terms of gene structure, prokaryotes and eukaryotes are very, very different. So in prokaryotes, the DNA sequence is directly maps to the protein sequence. And this may be like, well, duh, obviously it does, right? Just give me a second and we'll see how that doesn't apply in eukaryotes. So whatever the DNA sequence is, the protein sequence matches that directly, right? So if you have a mutation in the DNA here, you'll have a change in the amino acid here and so on and so forth. That's what these kind of lines and stuff are referring to. So that's called collinearity, right? The DNA sequence directly matches up to the protein sequence. So in some ways, prokaryotes are simpler in terms of gene expression than eukaryotes, some ways. Now for eukaryotes, however, that does not apply. And so the big difference here is eukaryotic genes have introns. Introns are non-coding gaps in the DNA. Well, not gaps. No, that's wrong. Hang on. Non-coding gaps in... How do I put that? And the transcript. So there's a lot more going on in a eukaryotic gene. And actually now we have some new words to learn, right? So exon equals coding sequence, right? Introns separate exons because they're in between the exons. Yeah. We have UTR, and that is untranslated region. So this is different from an intron. So introns aren't translated either, but introns are cut out of the mRNA before translation. The UTRs, one's at the 5' end and the other one's at the 3' end, stay as part of the mature transcript. Oh, it's still a bit too hot. Promoter, basically where RNA pol binds. And what's at the other end? Poly A. That's kind of like a 3' tail, oh, not trail, tail added to the mature transcript. So obviously there's a ton more stuff going on in eukaryotes with regards to gene structure than there are prokaryotes. And so we'll get into why this matters, later. We're not going to get into it in huge detail now, but you do need to remember that prokaryotes, the protein sequence directly corresponds to the DNA sequence. Eukaryotes, it does not. Right, so only these green bits here correspond to protein sequence. All the rest is not translated. And later on, we'll get into what these untranslated regions are used for. They're very important. Otherwise, they wouldn't be there, right? But we'll see what role they play. Okay, so now we've got a little bit of, idea about directionality of DNA. We've talked a bit about, the two different strands and also the actual structure of a gene, right? And this isn't all of it either. There's a lot more to the left and involved in initiation of transcription, right? Which we'll get to in a bit. But this is a pretty good overview. Now, one of the funky things is, though, right? These introns, and each eukaryotic gene has at least one intron, right? Guaranteed. Their size can vary from being kind of small, like 50 base pairs, like dinky tiny, to thousands upon thousands of base pairs, right? So that's one of the reasons why coding regions make up such a small part of our DNA, is because a lot of, even in genes, a lot of the gene is actually made up of space, introns, right? And so the average, human gene is about 27,000 base pairs long, right? But of that, only, 1,300 base pairs actually encodes for amino acids, right? So when we look at this picture, really what you'd want to do is you want to stretch that out, so it's probably, I don't know, the width of my couch, give or take, from like over there to there, and keep these green bits the same size. That'll give you an idea of how much just empty space essentially is in a eukaryotic gene. Now, I'm not saying that those introns don't serve a purpose, because they do. It's just they don't code for amino acids. Whereas prokaryotic genes, because they do not have introns, tend to be fairly small. I think E. coli has a genome of about 4,000 genes, give or take. And its genome is, what is it? I think it's in a couple of 100,000 base pairs, right? So prokaryotes have much more compact genes than eukaryotes do, or genomes rather. Okay, bit of a refresher. Differences between DNA and RNA. RNA has an OH at the second prime, so it's a ribose sugar. That's why DNA is deoxyribonucleic acid, because it has lost an O, hence deoxy. Whereas RNA is just ribonucleic acid. And here's actually, pardon me, something really funky about RNA. RNA can form really complex 3D structures. So that can be a mix of single and double-stranded RNA. So it's often, kind of taught that RNA is single-stranded, because as synthesized, it is single-stranded, right? It's not double-stranded like DNA is. But RNA forms the most craziest shapes. And that's through basically binding to itself, right? Forming double-stranded RNA. But the thing is, those double-stranded bits tend to be fairly short. Let's see if where's... So for example, this is what tRNA looks like, right? What it actually looks like, this is kind of like a conceptual picture. And this is a fairly simple one, right? I've seen stuff that looks like, I don't know, some kind of Escher diagram, right? Made up by someone who's been taking way too much acid and has been left alone in a dark room kind of thing. This is fairly simple, and still you can see like all of this. This is single-stranded RNA. We've got a little bit of single-stranded RNA down here, double-stranded RNA, which is where it's basically binding to itself. Right, so don't ever let anyone tell you that RNA is just single-stranded, right? Because it is not. It's actually in many ways a far more complex molecule than DNA is. DNA can form funky shapes, I guess, you know, but it's pretty dull compared to RNA. And actually, I was just reading the other day about coronavirus, right? So coronavirus has an RNA genome, it's an RNA virus, and actually has two large reading frames, like so. Its genome consists of two large reading frames. Each one gets turned into multiple proteins, right? It's kind of similar to prokaryotes in that way. We'll get to that later. But they're basically kind of part of the same bit of DNA, but they're in alternate reading frames. And so it actually causes a jump from one reading frame to the other through the formation of what's called a pseudo knot. I don't know what that is. It's like a knot that isn't a knot, maybe. It's not a knot. Anyway, that's a bad joke. You shake your head now, Robert, but you'll be telling jokes like this when you get older, I'm sure. Maybe, maybe not. Anyway, and so like the actual structure of the RNA genome in coronavirus is important in translating that genome into protein. So RNA is pretty wild. It's arguably one of the most interesting molecules there is. Okay, so back down to kind of Earth. Obviously, RNA is made by RNA polymerase, right? Easy again, way to remember what an enzyme is just add ASE to the end of the word of the thing that it makes. So peptidase, actually, no, that's not quite right. Peptidase cuts up peptides. RNA polymerase polymerizes RNA polymers, right? And the key thing here is, again, that RNA nucleotide can only be added to the 3' end of the newly made or making RNA strand, right? So if we have our DNA template strand here, right, again, the coding strand is going to be over to the left, right, over here, isn't shown, right? But the coding strand's going down the page, template strand is going up the page. So RNA polymerase catalyzes the bonding of a new nucleotide, right? Base pairing with the template strand. And this is all kind of, it's sort of transient, right? So we've got this base pair complementarity, right? We've got a DNA RNA hybrid, basically. But this is like a zipper that doesn't work very well. I don't know if you've ever had those, you know, it kind of zips for a little bit and then it breaks apart afterwards. That's kind of what this DNA RNA hybrid is like, right? So it base pairs for the region that's being synthesized, and then up there, the bit that's already made dissociates from DNA, right? So we don't get like a long stretch of DNA which has RNA bound to it, right? That binding only occurs for a small part, and then that RNA kind of leaves and the DNA closes up behind it. But anyway, this nucleotide comes in. This is a uracil, right? So it matches an A in the template strand and it corresponds to a T in the coding strand. That's brought in, base pairs with adenine, and then RNA polymerase synthesizes the phosphodiester phosphodiester bond, sorry, between the phosphate on the incoming nucleotide and the 3' OH on the one that's already there. And that reaction is powered by the energy released in removing this diphosphate, this pyrophosphate. All right, how are we doing so far? Anyone need any clarifications, or what did you just say? Cool. Okay, so there's 3 different RNA polymerases there. And essentially each one is a whole bunch of proteins and other stuff together. Right? It's called a holoenzyme. Right, and holoenzymes are typically enzymes with like some kind of cofactor. I'm not sure what that is in RNA polymerase. But these are large, complex, multi-protein proteins. So bacteria only have 1 RNA polymerase. This makes it a lot easier. Eukaryotes have 3, at least 3, and maybe another one as well, which I can't remember off the top of my head. RNA polymerase 2 transcribes all of the mRNAs, as well as the small nuclear RNAs, which we'll get to next week. Schnurps. RNA pol 1 does the ribosomal RNA. RNA pol 3 does the tRNAs, which there are 20. And the small component of ribosomal RNA. So there's actually 2 different ribosomal RNAs in every ribosome. So they're kind of specialized as to what they synthesize. And presumably, although I don't actually know this for sure, but presumably there's going to be differences in the DNA which they bind to, which will direct them to those specific genes. So RNA pol 2 doesn't bind to genes encoding ribosomal RNA, for example. You'll go up. And so this is what RNA pol 2 looks like more in like cartoon version based on its structure. And so essentially it has kind of an entry and an exit point for the DNA, and an entry point for the nucleotides, the RNA nucleotides coming in, and for the RNA, the newly synthesized RNA coming out. Right, so you can tell direction here. Not only does it tell you where the DNA enters, but it also shows you the 3' end of the mRNA. So you know that it has to go from left to right in this picture. Now, one of the funky things about RNA polymerase is it physically bends the DNA, sorry. So you remember back to the initiation of DNA replication, those initiation proteins that bind and then they bend the DNA and it kind of pops it open, forms a bubble. So you have single-stranded DNA. That's essentially what RNA polymerase does, right? By bending the DNA here in this kind of almost 90-degree bend, it physically forces the double helix open into a bubble. And that bubble allows RNA nucleotides to come in and base pair with the template strand. And this RNA polymerase will process along the DNA, right, like a little motor. And it will keep that bubble open with it as it goes along. So the bubble doesn't stay open. As soon as the DNA exits the RNA polymerase and is kind of free to move around, it will twist and close back up again. And so this isn't like a replication bubble where you have an ever expanding bubble, you know, between the two original strands. This is just a transient, temporary deal, just to allow this short-term base pairing between the RNA nucleotides and the DNA molecule. Now, as with everything, like there's all these molecular processes have a beginning, a middle, and an end. And the middle is actually often the least interesting part, by and large. But the same is the case for transcription. So we have initiation of transcription, right, which is actually an astonishingly complex process, particularly in eukaryotes. We have transcription, you know, which is just making the RNA. And then we have termination of transcription. So those are the kind of the three things that we need to cover before moving on to RNA processing in eukaryotes. Right, and so that's really what the rest of this PowerPoint is about. And we'll get through as much of it as we can today, and anything we don't, we'll continue on Tuesday. So in bacteria, it's actually kind of fairly straightforward in a sense, right? They're much more conserved in terms of how transcription is initiated. And so this promoter word, right, which you come across again, this is essentially a sequence upstream, right? If we're going from left to right, which we are here, this is further to the left, right? This is a piece of DNA which can be huge, particularly in eukaryotes, which does not encode protein. It's responsible for the binding of RNA polymerases, transcription factors, and anything else that is necessary to begin transcription. So these can be very small, particularly in bacteria. They can also be huge in eukaryotes, depending on the complexity of the control of transcription. And they will always end with this little red arrow, or it's just going to be an arrow of some sort. That denotes the start of transcription. So one of the key things to remember, and this is universal for all organisms, RNA polymerase is not very good at binding promoters on its own. It needs help. And so there will typically be other proteins which recognize promoters, and they kind of recruit or guide RNA polymerase to those promoters to begin transcription. So RNA polymerase on its own is a fairly dumb processing machine. It's very good at making RNA, not very good at anything else. So it relies on other proteins, transcription factors, for example, to tell it where to go to do its job. And what you'll find in promoters is there are going to be sequences in the DNA which are common to multiple promoters. These are called consensus sequences. Right, what we mean by a consensus sequence, you can kind of graph it out here. Right, which base are you more likely to find at any given position? So some bits of DNA, like, position 18, 21, you're fairly equally to find any particular base. Right, so most likely those positions are not important. Because if they were, there would be a selective pressure to weed out mutations or changes in those bases. Other bases, you'll only ever, like 16 and 17, find that base there, or other positions, sorry. These are really important because if you change that organism most likely doesn't survive or isn't very fit. And so that mutation disappears. So these bases where there's only a single base or one very common base, those are going to be very important in whatever function that sequence is responsible for. And so that's what we mean by a consensus sequence. Right? If you look at a whole bunch of different genes or different organisms or whatever, and you find in every one of them at position 17 relative to whatever else we're talking about, there's a G, then that G is very important. It's recognized by something. And if that G isn't there, then that thing doesn't recognize it, basically. Right? And so when you look at bacterial promoters, they have a very clear, they've got two consensus sequences, -10 and the -35 sequence. These are what are recognized by RNA polymerase and the sigma transcription factor. If you don't have these sequences, you don't get transcription. And so this is called the Tartar box, or Tartart, if one were to, pronounce it. And there's a similar one in eukaryotes too. And there's another one further downstream, or upstream actually is probably the better term. And how close, so if you look at like through these sequences, these are different genes. There's some lac genes of the tryptophan gene, galactosidase, recombinase, so on and so forth, right? And the spacing is approximately -10 and -35. So minus meaning upstream, right? Prior to the transcription start site. So if you look through these, you'll see that they don't all match, right? This one down here, recombinase gene, matches perfectly. The one for LAC-I is so-so. So how close these sequences are to the consensus also determines how strongly they're recognized by and bound by proteins. So you can have different strength promoters as well. And obviously mutations in those sequences, if they change them to be closer to the consensus, that promoter will become stronger. You'll have more transcription from that promoter. If they change them further away from the consensus, then you'll get less transcription because you'll have a weaker promoter. Right, so those mutations can have fairly large effects on the actual organism itself. Now, in terms of how it works, if we're to kind of draw out the gene here, right, so kind of to the right, after that, actually, let's embigorize this. There you go. Right, after this arrow to the right is the gene itself. Well, the coding region, let's say. This is all part of the gene. So the promoter is part of the gene, even though it isn't translated into protein. And so you have this promoter, which is just upstream of the transcription start site. And you have two kind of key players, right? You have the RNA polymerase, which is that holoenzyme, whole bunch of different proteins. And then you have a sigma factor, right? And there are other transcription factors too, which will guide RNA polymerase to specific genes based on different conditions, like stress or high temperature or low nutrient availabilities, things like that. The sigma factor is like the generic one, essentially. And so its job is to recruit RNA polymerase to the promoter. RNA polymerase binds to the promoter using particularly the -35 sequence. And once it does, I mean, it stays complexed, it pops open the template pops open the strand, right? So I don't know if you remember me talking earlier about T's and A's have fewer hydrogen bonds, right? What do we find in the promoter? Lots of T's and A's, right? And so this is a region which is going to be easily popped open because there are less hydrogen bonds holding it together than elsewhere, right? So it's just kind of neat to think about that, kind of go back to it. Right, so that region is popped open, nucleotides come in, and RNA polymerase synthesizes the new strand of RNA, which you can see kind of here. And it will process along the DNA, taking that little bubble with it, so you can actually see it's moved from the promoter into the coding part of the gene itself. And it will synthesize a mRNA that matches the coding strand using the template strand at the bottom. So that's all pretty straightforward, right? It's fairly, you know, basic in a sense. It's a lot more complicated in eukaryotes, as always, typically. Okay, but before I get further on, I want to make sure that, you know, everybody's up to speed with what we're talking about so far. Does anybody have questions about any of this? Or even, you know, what's my favorite type of coffee, you know, stuff like that. Usually, as long as it's hot. So this is where we start getting into those detail-oriented processes, right? You need to know what sigma factor is because it's super important. It's hard to think of sigma factor as being something other than just a name, right? Because it's a very abstract concept. But it's, if you think about what it does, right, it's the guide, right? It's what guides RNA polymerase to the promoter to initiate transcription. So sometimes I often find thinking about what things do is a better way to remember what they are. Okay, everybody all up to speed? Again, this is stuff that you'll need to go over probably a few times for it to properly sink in. Might not sink in straight away, and that's normal. But do make sure, you know, go over. There's going to be a lot of language too, a lot of different words that we'll be using. So in eukaryotes, it's really not as well defined, right? So promoters are much, much bigger, right? And much more complex. And actually we have, I'm going to very quickly use this diagram. There. In eukaryotes, we have now two types of, well, not two types of promoter, two parts of the promoter. We have what's called a core promoter. And I'm going to be lazy and shorten eukaryotes to euks. And that's really where RNA polymerase II binds. Plus the general transcription factors. And that can be, I don't know, a few hundred bases, maybe. Right. And so it's a core promoter, right? And so what that's telling you is that that's the bare minimum necessary for transcription. It's not saying that transcription is going to work very well, or you'll get a lot of transcription, but that's the bare minimum. Without that, it's not going to work at all. And then you also have a regulatory promoter. And this is where all the other transcription factors and transcriptional activator proteins and repressors bind. And that can be thousands of base pairs long. Sometimes even millions. And it doesn't have to be continuous either. There can be regions like way elsewhere in the chromosome that have an important role in the activation and regulation of that gene. And so transcription initiation in eukaryotes is far more complex than prokaryotes. So in eukaryotes, we have what's called a TATA box, T-A-T-A. Right, it's smaller. It's usually around 25 to 30 base pairs upstream of the transcription start site. It's much less tightly conserved. But this is really important because it's recognized by the TATA box binding protein, very imaginatively named. And so as with prokaryotes, RNA polymerase II cannot bind DNA on its own. Again, I don't know, sounds a bit kind of snarky, but say it's dumb as a box of bricks. But it doesn't decide what genes are turned on or not. And doesn't even know where to go, right? It's kind of blind. And so there are 6 other proteins. These are called general transcription factors. And so these are involved in transcription of all genes. And then you have gene-specific TFs. And those are responsible for transcription of a set of genes. For example, all those involved in heat shock response. And we'll get into more of that when we talk about control of gene expression in the next session. So these are all transcription factor 2, A, B, and D, E, F, and H. And so what does that actually look like? So let's kind of blow this up again. And so, too far. So essentially what you have is your core promoter, your regulatory promoter, and your transcription start site. In that core promoter, you'll have the TARDA box. That's what's recognized by the TARDA box binding protein. And that complexes with one of the general transcription factors. And so this is basically the, hello, I'm here, come bind me sign for RNA polymerase and all the other stuff. So this is like a kind of a scout that goes ahead to find the right spot. So once TBP and transcription factor 2D bind to the TARDA box, that causes everything else, RNA polymerase 2, right, which again is a large multi-unit enzyme. The other 5 transcription factors, and so these are also involved in kind of retaining RNA polymerase so it doesn't wander off. And another protein called mediator. And what that does is it kind of acts as an intermediary between RNA pol 2 and other proteins, e.g. the transcription activator proteins. Now, even with all of this assembled, you still don't have transcription, or at least not very effectively. So now you need to kickstart this, literally give it a kick up the butt. And that's done by the interaction of other proteins, those transcriptional activator proteins, with the mediator. And quite typically that involves bending the DNA so that proteins bound elsewhere to it, for example, over here, are able to interact with the mediator. So again, in prokaryotes, it's really quite simple. Sigma factor binds the promoter, recruiting the core RNA polymerase. As long as those two bind to the promoter, transcription begins. So prokaryotes have other ways of controlling whether or not transcription occurs. It's not at the level of the RNA polymerase binding. That's pretty much kind of like an auto thing. Right? In eukaryotes, it's very different. So we still have the kind of binding of the what's called the basal transcription apparatus, like the basic set. That's mediated by tidal box binding protein and TF2D. So in a sense, these two play the same role as sigma factor does in bacteria. Kind of analogous. So once these two are bound, all the other stuff is now recruited. And so far, relatively similar, I guess. You can kind of see the parallels. It's after that that things get very different, right? So if there are no transcriptional activator proteins present, you may get some transcription. From that basal transcription apparatus being there. Yeah, sometimes it just does. However, it's not going to be very effective. It's not going to happen a lot. And so in eukaryotes, you need other proteins, not just the ones involved in recruiting RNA polymerase, but you need other proteins to activate it. This whole jazz on its own doesn't start itself very reliably. It doesn't leave, right? So it's stuck there, but it's not going to initiate transcription on its own very reliably. And so in eukaryotes, you need other proteins, these transcriptional activator proteins, co-activators. You need other bits of DNA for proteins to bind to. That's the regulatory promoter. And so that's what controls whether or not transcription occurs. And so that's a key difference between eukaryotes and prokaryotes. Prokaryotes, as long as sigma factor and RNA pol binds, there's nothing stopping transcription in and of itself, right? It will begin transcription on its own. In eukaryotes, that isn't enough. Sometimes it is, it's just not very reliable. You need other proteins to start the process. And so now you can see how you can interfere with that process. Let's say we have other proteins that bind to these regions but don't activate RNA polymerase. They don't interact with mediator. Now you can actually stop transcription from occurring by inhibiting with that process or interfering with it. Right, so now we get a lot more complexity. The main take home is that this regulatory promoter, and again, this can be a long, long way from the actual gene itself, contains sequences that are bound by proteins that are important for activation of transcription. Without them, you don't get very much transcription. All right, I think that's a good a place as any to stop for the day. Anyone have questions so far? I'd strongly recommend going over all of this over the weekend, fun as that might be, and making sure that this stuff sticks. If you have any questions, bring them on Tuesday and we can go over stuff then. Oh, actually, no, hang on. I forgot. Doesn't finish at 1/4 past. Finishes at 1/2 past. Yay, we got another 15 minutes of class. Woohoo! I can hear the excitement and jubilation through my computer screen. Okay, and normally I'd go like, oh yeah, well let's just can up for the day and we'll have an earlier lab, but we do need to get through a bunch of stuff because RNA processing and translation are also fairly solid topics. Sorry about that. Gave you a false hope, which is never fun. Yeah, sorry, Marcus. Sad face. Oops. Okay, so that's initiation, right? That's getting things going. The actual bit in between, the elongation, is pretty straightforward. It's simply synthesizing the mRNA strand or the RNA strand. Oh, I feel really bad about that now. Sorry, that wasn't intentional. But you got to stop at some point. And so again, RNA polymerase in and of itself, it doesn't know when the gene ends. Doesn't know really where it starts, right? It requires other stuff or other information or other proteins to tell it when to begin and when to finish. And so this is when to finish is important, right? Because if you imagine we've got, you know, multi-million base pair chromosome, you don't want to transcribe the entire chromosome, right? That would be a huge waste of energy, right? And time, and it'd just be bad idea in general. So we need some way of stopping this, right? We need to be able to kind of cut short RNA polymerase before it starts wandering off into the boonies and just transcribes everything that it can find. So in bacteria, we've got two ways of terminating transcription. In eukaryotes, it's again not quite as straightforward, right? And so in bacteria, we have what's called intrinsic termination. That's all about information that is intrinsic to the sequence that's being transcribed. Right, so there's actually something in the sequence of the gene itself which tells RNA polymerase to stop. And then we have something called Rho-dependent termination, and Rho is a protein which is involved in the termination transcription. And for all of these, right, intrinsic, Rho, and not so much for eukaryotes, but in bacteria, this all relies on the formation of an RNA structure. And that structure is called a termination loop. So in intrinsic termination, we have that termination loop, right? And so it basically looks like so, right? It's where the RNA base pairs with itself and forms what's called a hairpin loop or a hairpin structure. It also relies on forming a series of uracils. And so again, this is information encoded in the gene itself. And so when those U's are transcribed, essentially that slows down the RNA polymerase. And what that does is it allows time for this termination loop to form. And really, it acts like a lever. Right, and so if we go back here, so essentially, and this is RNA pol 2, but it's similar enough to bacterial RNA polymerase that it'll work. Basically, what happens is that RNA polymerase pauses after it makes that string of uracils, and that allows the RNA that's partly outside of the enzyme to form this loop, and it kind of forces its way into the channel in which the DNA is present. And it pretty much just pushes the RNA polymerase off of the DNA. Right, so you can kind of see there's actually like not a lot of space in there, right? It's a very specific kind of three-dimensional shape. And the formation of that RNA structure inside there is going to push other things out. And that's how intrinsic termination works. So again, there's a series of A's in the template strand. Those get transcribed into uracils. That slows the RNA polymerase down and gives time for this termination loop to form, which forces the RNA pol off the DNA. So that's intrinsic termination. Extrinsic termination, or Rho-dependent termination, relies on a protein called Rho. And so we again have a terminator sequence, but now we need a second thing, a protein, to come along and force the RNA polymerase off of DNA. And so we have this kind of setup, right? So here's our RNA polymerase. Here's our little transcription bubble inside. And we have what they mean by unstructured RNA is RNA that doesn't form loops and hairpins and other secondary structures. Right, and so that allows this protein to bind. Right, essentially, this is like a little molecular motor, and it immediately starts chasing after RNA polymerase. Like RNA polymerase is in the distance, and Rho is like running after it down the mRNA strand to try and catch up. Now, normally, those two are fairly matched in speed, so Rho does not catch up. We need something to slow down the RNA polymerase. And that something is one of these terminator loops. So similar to what we see here, but this terminator loop doesn't form inside of the RNA polymerase. But what it does is it causes RNA polymerase to pause. And that pause allows Rho to catch up. And as soon as Rho catches up, it essentially unwinds the RNA-DNA hybrid. This bit up here, let's kind of configurize this. Right, so this hybrid here between the mRNA that's being synthesized and the DNA template strand. Once Rho catches up, it moves into the RNA polymerase and forces that RNA-DNA hybrid apart and Rho off, and Rho RNA polymerase off of the DNA. And obviously, as soon as RNA polymerase gets hoofed off of the DNA, transcription stops. Now, eukaryotes is much less well defined, but there is one mechanism that we know of, at least, that's involved in transcription termination. Now, because eukaryotes process their RNA, we actually have a step that is involved in that. So in eukaryotic mRNA, we need to stick a sequence onto the end of the new transcript to protect it, basically, from RNases, from degradation. And so in eukaryotes, the RNA polymerase just keeps going well past the end of the actual coding sequence. Actually, well past the end of even the 3' UTR. And essentially the new mRNA, it's called a pre-mRNA because that stands for precursor. And that's not ready for translation. Dang it, can't see what I'm typing. And so after the 3' UTR, there's a site that's recognized by an endonuclease. And that RNA is cut. And so that releases the pre-mRNA that can then continue being processed before being exported to the cytoplasm. And RNA polymerase just keeps going on, oblivious, essentially. It's like, yeah, I'm having fun. Running down the track. But now we have a free end, right? And RNases typically attack RNA from the ends. Not always, as in the case of this, cuts in between, but typically RNase is like free ends of RNA, juicy, tasty. And so when that free end becomes available, that's recognized by another RNase called RAT1. And so RAT1 is going to chew up that kind of useless bit of RNA that the RNA polymerase is still making, right? So this bit of RNA isn't going to be used for anything. It's surplus, right? It's kind of not pointless, but it's not going to be useful. And so similar to Rho, right? So Rho plays a kind of a catch-up game. Rat1 also plays a catch-up game. And its job is trying to catch up RNA polymerase before RNA polymerase transcribes too much DNA. And as soon as the Rat1 reaches the RNA polymerase, it kind of forces its way in. And as with Rho, kicks off the RNA polymerase from the DNA. So transcription is terminated. So in eukaryotes, there's a bit more waste involved, right? So RNA polymerase makes a bunch of RNA which isn't going to be used. But that cleavage is really important for subsequent RNA processing. So it kind of has a benefit in that sense. All right, and now we have actually come to the end of the class. Sorry about that extra 15 minutes. But that also allowed us to get to the end of this so we can start fresh on Tuesday, which is not a bad thing. So we have time to get through everything we need to then. Cool, so I'm gonna call it a day now. We're gonna meet back at 2 o'clock for lab, which is gonna be all about DNA quality control, essentially, from the DNA purification. And so we'll meet back at 2. All right, a little bit kind of mind blending, but yeah, lots of stuff to take in. All right, everybody, I see you soon. Enjoy your lunch, and I'll see you at 2.